Dear students, this video lesson is for first year BSc, first semester in the subject Food Science and Nutrition, paper Human Physiology. The topic is composition of blood, presented by Dr. Rajeshwari. Blood is a fluid connective tissue that consists of plasma, blood cells, and platelets. It circulates throughout our body, delivering oxygen and other nutrients to various cells and tissues. It makes up 8% of our total body weight. On an average, adult possesses around 5 to 6 liters of blood. The specific gravity of blood is 1050 to 1060. Viscosity of the blood being 4 to 5 times of that of water. Blood is slightly alkaline in nature by having pH of 7.4. Blood contains different types of cells. These cellular elements of blood accounts for about 45% of the total blood volume. It is called as PCV or packed cell volume or hematocrit. It includes erythrocytes or Wbcs or red blood corpuscles, which account for about 4 to 5 million cells per cubic millimeter of blood. Leukocytes or Wbcs are white blood corpuscles. The normal count of these is 4,000 to 11,000 per cubic millimeter of blood. The platelets in the blood are also called as thrombocytes. They account for about 1.5 to 4 lakhs, on an average 2.5 lakhs of platelets per cubic millimeter of blood. Blood is a highly specialized circulating tissues consisting of several types of cells. The 3 types of cells suspended in a fluid medium known as plasma. Plasma contains amino acids, nutrients such as proteins, albumins, fibrinogen and globulins, nitrogenous waste materials, some electrolytes and dissolved gases like oxygen and carbon dioxide. The formed elements in the blood are the blood cells, RBCs and WBCs, and also platelets. Leukocytes again categorized into different categories as granulocytes and agranulocytes. Under granulocytes, there are 3 types of WBCs named Neutrophils, Basophils and Isonophils. Agranulocytes are lymphocytes and monocytes. Blood plasma is a yellowish amber color or straw color liquid component of the blood that holds the blood cells in suspension. It is the liquid of the blood that carries blood cells and proteins throughout the body. It makes up about 55% of the body's total blood volume. It is the intravascular fluid part of the extracellular fluid, that is, all body fluids outside the cells. Plasma is mostly water, up to 95% by volume, and contains important dissolved proteins up to 6 to 8% called serum albumins, serum globulins and fibrinogen. Apart from that, glucose, clotting factors, electrolytes, hormones and dissolved gases like oxygen and carbon dioxide are present. The water of plasma is freely exchangeable with that of body cells and other extracellular fluids and is available to maintain the normal state of hydration of all tissues. The major solute of plasma is heterogeneous group of proteins constituting about 7% of plasma by weight. You might have heard of serum, blood serum. The difference between blood plasma and blood serum is that plasma contains clotting factors, blood serum has no clotting factors. Now let us learn how does the plasma functions in the body. One of the plasma's main function is the removal of wastes from cellular function that help to produce energy. Plasma accepts in addition to transporting waste and regulating body temperature, plasma has several other key functions that are carried out by its different components such as proteins, electrolytes, and immunoglobulins. Plasma contains two key proteins called albumin and fibrinogen. Albumin is vital for maintaining a balance of fluid. It is called as oncotic pressure in the blood. This pressure helps to maintain the colloidal osmotic pressure across the capillary wall. Normally it is 25 to 30 mmHg. This pressure is what keeps fluid from leaking into areas of the body and skin where less fluid usually collects. Osmotic pressure across the capillary wall can be exerted both by the crystalloids, example urea, sodium and glucose, and the colloids such as plasma proteins. However, capillary wall is completely permeable to the crystalloids. Therefore, crystalloids hardly contribute to capillary osmotic pressure, and proteins exert an osmotic force of 25 mmHg across the capillary wall. Osmotic pressure, that is what is created here, is inversely proportional to the molecular size and shape, and it is directly related to the concentration of molecules. Therefore, 80% of osmotic pressure, colloidal osmotic pressure, is due to the albumin because of least molecular weight, that is the molecular size, and maximum concentration. Colloidal osmotic pressure across the capillary wall helps to maintain the exchange of fluid and tissue level at tissue level. The rate of fluid exchange, that is filtration, absorption, at any point along a capillary depends upon the balance of forces, which is called Starling forces. Hydrostatic pressure at an arteriolar end is 37 mmHg. Therefore, some fluid is forced out of capillary bed and becomes 12 mmHg. Leukocytes can manufacture a few substances called triphones or chiral from plasma proteins which help in the nutrition of tissues. Fibrinogen help to reduce active bleeding, making it important part of the blood clotting process. Immunoglobulins of plasma contains gamma globulins It's a type of immunoglobulin which helps the body fight off infections. The electrolytes of plasma conduct electricity when dissolved in water, hence their name. Common electrolytes of plasma include sodium, potassium, magnesium, and calcium. Each of these electrolytes play a key role in the body. The plasma proteins also carry immune functions. Gamma globulin produce antibodies which provide immunity to the body. Transport function of plasma proteins include the hormones, which are these proteins combine loosely with many agents like hormones such as thyroxine and cortisol. They combine with drugs and some metals. They also play a reservoir function. Plasma proteins form loose bond with the hormones, drugs and metals to serve as a reservoir from which the same are released slowly. This plays a beneficial role during the starvation. This is about the plasma and its function. Now, let us move on to the structure and function of WBCs or white blood cells. WBCs are also called as leukocytes. Unlike RBCs, they are nucleated and independently motile. The number of white cells in normal blood ranges between 4,000 to 11,000 per cubic milliliter of blood. They are of two types, namely granulocytes. In them, the cytoplasm of granulocytes having granules, and agranulocytes where the cytoplasm does not contain granules. Further, granulocytes can be distinguished into neutrophils, eosinophils, and basophils. The granules in the granulocytes lineage have different affinity towards the neutral acid and basic stains, giving cytoplasm different colors like blue, purple, and pink. The agranulocytes are the lymphocytes and monocytes. The granulocytes contains neutrophils, eosinophils, and basophils. The granules in the granulocyte lineage have different affinity towards these, the basic and the major type of WBCs are neutrophils. They are the most abundant type of WBCs are granulocytes and make up 40 to 70% of all white blood cells. They have a diameter of 12 to 15 micrometer. Their nucleus is divided into 2 to 5 lobes connected by a fine nuclear strand or filament. Human neutrophils have an average diameter of 8.85 micrometer. The cytoplasm is transparent due to the presence of small granules. With the eosinophil and the basophils, neutrophils form the class of polymorphonuclear cells named for the nucleus multilobed shape. The nucleus of these cells has a characteristic lobed appearance. The separate lobes connected by a chromatin. The nucleus disappears as the neutrophils mature, which is something that happens in only a few other types of nucleated cells. The cytoplasm, the Golgi apparatus, is small. Mitochondria and ribosomes are sparse. The rough endoplasmic reticulum is absent. The cytoplasm also contains about 200 granules. The average lifespan of inactivated human neutrophils in the circulation has been reported by different approaches to be between 5 and 135 hours. Upon activation, neutrophils marginate, that is, position themselves adjacent to the blood vessels endothelium and undergo selection dependent capture followed by integrin addition, dependent addition in most cases, after which they migrate into tissues where they survive for 1 to 2 days. Other functions of WBCs. Apart from this, neutrophils also have these functions: chemotaxis, antimicrobial function, phagocytosis, and degranulation. Chemotaxis is bacterial products interact with plasma proteins to produce agents that attract neutrophils to the site of injury or inflammation called chemotaxis. As a result of this, neutrophils adhere each other, forming clumps and become immobile. The chemotactic agents include components of complement system, leukocytes, polypeptides from lymphocytes, basophils, and mast cells. They are the other WBCs. Once the microbes found in the blood, the neutrophils become active. They become antimicrobial in function. They prevent the activity of microbes. The primary function of neutrophils is the phagocytosis, the ingestion and destruction of microorganisms or other foreign particles. For this reason, neutrophils also called as phagocytes. When a neutrophil is faced with a microorganism or foreign particle, phagocytosis begins. Phagocytosis is coated bacteria when bind to the receptors on the neutrophil cell membrane and get phagocyted by a process of endocytosis forming antigen-antibody complex. This way, neutrophils also phagocytize many foreign inanimate substances, for example, some carbon particles or sodium urate crystals, etc. Phagocytosis occurs at pH between 6 to 8 and it is not dependent on the oxygen. It is aided by bivalent cation and is prevented by calcium chelating agents. A neutrophil can phagocytize 5 to 20 bacteria before the neutrophil itself becomes inactivated and dies. Macrophages are more powerful phagocytes than neutrophils. They can phagocytize as many as 100 bacteria, can engulf particles up to the size of a RBC and have the ability to phagocytize necrotic tissues and even dead neutrophils. Degranulation is after phagocytosing the bacteria, that is coating the bacteria, bactericidal substances, these substances are also called as defensins such as lysosomes and peroxidases are released from the lysosomal granules into the digestive pouch, that is called degranulation. where they kill and digest the bacteria. Apart from this, release of lysosomal enzymes called like histamine and 5-HT into extracellular fluid produce inflammatory responses. This is about the function of neutrophils. Now, the next granulocyte is eosinophils. The eosinophils are quite rare in blood. They have the same size as the neutrophils and generally have bilobed nucleus. Eosinophils, sometimes called eosinophiles, or less commonly acidophils, because they take up acid dye into their cytoplasm, and one of the immune system component which is responsible for combating multicellular parasites and certain infections. Eosinophils function include movement to inflamed areas, trapping substances, killing cells, antiparasitic and bactericidal, that is, killing of bactericidal activity, participating in immediate allergic reactions, and modulating inflammatory responses. As you can see in this picture, the eosinophils has the diameter of 10 to 14 micrometer. Nucleus is purple color. These cells usually bilobed. The two lobes are connected with the chromatin thread, thus producing spectacle appearance. Cytoplasm is acidophilic. Therefore, it appears light pink in color. The cytoplasm is granular. The granules being coarse, stain bright red with acidic dye, that is called eosin. Granules do not cover the nucleus. They contain very high peroxidase content, lysosomes, most of the enzymes found in the neutrophil granules. Eosinophils also take up mild phagocytosis, that is, killing of the cells of bacteria. Eosinophils collect at the site of allergic reactions and limit their intensity. They enter the tissues and are especially abundant in the mucosa of respiratory tract, gastrointestinal tract, and urinary tract, where they provide local mucosal immunity. Eosinophils attack parasites that are too large to be engulfed by phagocytosis. Eosinophil granules release chemicals which have toxic to larvae of parasites. That chemical is peroxidase. The next type of granulocytes, which account for about 0.5 to 1% of WBCs, are basophils. Basophils are the rarest leukocytes, having a diameter of 9 to 10 micrometer. Nucleus is as in eosinophils. Cytoplasm is slightly basophilic, therefore appears blue and granular. Granules are coarse, stains purple or blue with basic, that is, methylene blue dye. Granules are plenty in number and overcrowd the nucleus, resulting in obscure boundary of the nucleus. Certain histamine and heparin are present. The granules also contain eosinophil chromatic factor of anaphylaxis. The functions of basophils. include the keeping the immune system functioning correctly. Mild, they possess mild phagocytosis, liberate histamine and extracellular factor A, which leads to allergic manifestations. Extracellular factor A attracts eosinophil into the region where antigen IgE interaction. Extracellular fluid A is a chemical mediator of immediate hypersensitivity reactions. These ranges from mild urticaria, that is localized skin allergic reactions, to severe anaphylactic shock. Basophil cytoplasm liberates heparin, which act as anticoagulant and keeps the blood in fluid state in the body. It activates a hormone lipoprotein lipase, which facilitate absorption of triglycerides after meals. Now let us learn about the agranulocytes. They are lymphocytes. Lymphocytes are 8 to 14 micrometer in diameter, and generally they are smaller than other leukocytes. Lymphocytes include natural killer cells called T cells and B cells. Lymphocytes are also of two types. Large lymphocytes, their diameter is usually from 10 to 14 micrometer in diameter, which are the precursors of small lymphocytes. Small lymphocytes are also called as B cells or B type cells, having the diameter of 7 to 10 micrometer, and they are responsible for antibody production. Both large and small lymphocytes have the same structure, as you can see here. The picture on the left hand side is the neutrophil. Lymphocytes make up between 18 to 42% of circulating leukocytes. The nucleus of the lymphocyte is single, very big. You can see in the picture, purple in color. The shape of the nucleus is round, oval or indented. Centrally positioned and occupies whole of the cell, leaving marginal cytoplasm at one end of it or all around it. Nuclear chromatin is coarse and lumpy, that is shapeless. Cytoplasm is pale blue, you can see in the picture, scanty. Its amount is always less than the amount of the nucleus. The function of lymphocytes include: they are the cells that specially organized and respond to foreign antigens and are mediators of humoral and cellular immunity. Lymphocytes, the unique cells of adaptive immunity, are the only cells in the body that express clonally distributed antigen receptors, each specific for a different antigenic determinant. Lymphocytes play a major role in both cellular and adaptive immunity. Lymphocytes produce antibodies, that is, immune substances, especially in delayed hypersensitivity, etc. The next type of agranulocytes are the monocytes. Monocytes accounts for about 1 to 2%. They are the largest leukocytes, having a diameter of 16 to 20 micrometer. Most monocytes are the precursors of macrophages, which are the larger blood cells. They can differentiate into macrophages and myeloid lineage dendritic cells. As a part of the vertebrate innate immune system, monocytes also influence the process of adaptive immunity. Monocytes are amoeboid in appearance, you can see in the nucleus, and have non-granulated cytoplasm. They have a pale staining characteristic, having single nucleus, round and indented, that is, the nucleus is kidney shaped most of the time, eccentric in position, that is, present on one side of the cell. You can see that in the picture. Nuclear chromatin is finely reticular. Cytoplasm is usually pale blue and clear. Granules sometimes contains fine purple dust-like granules called azure granules, which may be few or numerous. The main function of the monocytes include active phagocytosis. Monocytes follow the neutrophils in the areas of infection and inflammation and constitute a second line of defense. Monocytes enter the circulation from the bone marrow, but after 72 hours, they enter the tissues to become tissue macrophages. All tissue macrophages come from circulating monocytes. Lifespan of monocytes is approximately 3 months. Monocytes may also kill tumor cells after sensitization by lymphocytes. Monocytes synthesize complement and other biologically important substances like prostaglandin E and clot promoting factor. The monocytes and their macrophages and dendritic cells progeny serve three main functions, as I told you, in the immune system. These are phagocytosis, antigen presentation, and cytokine production, that is, production of antibodies. That ends the structure and function of WBCs. Thank you.